Welcome viewers. Today in this session we will be talking about the different types of DNA modification enzymes present in bacteria. We will be mainly talking about on two main types of DNA modification enzymes are DAM and DCM. We will be talking about what is DAM, what are the different physiological functions of DAM, similarly what is DCM, how does it help in playing the different cellular processes. DAM and DCM are a part of DNA modification enzymes first or found to be associated with restriction modification system. The modification enzymes are those kind of enzymes which covalently modifies the DNA bases. DAM and DCM are a type of Methyltransferase enzyme. With the help of these kind of enzyme, the regulation in gene activity in terms of replication, transcription, DNA repair processes are modified or they are controlled. It is a kind of epigenetic regulation occurring in the living world ranging from prokaryotes to the eukaryotes like us.
Ma'am, I've got a question. What does the term epigenetics actually mean?
The term epigenetics refers to any change in gene activity which is heritable in nature, and the change in gene activity is not caused due to the change in base sequence but due to the modification of bases. Now let us first see why DNA methylation is absolutely essential in prokaryotic system. With different kind of research work, it has been found that the DNA methylation is helpful in playing different physiological roles. It plays a very vital role during DNA replication, during sequestration of the nucleoid into the dividing cell. It plays a very important role in DNA repair system, as well as we already know that DNA modification is an important part of restriction modification enzymes. Besides this, it also has a determinant factor in playing or controlling the bacterial virulence and henceforth the pathogenesis. So in this way, the modification enzymes are very important. Now, with further development, it has been found that the methyltransferase reaction, so as the name suggests, so it must involve a transfer of the methyl group. The transfer of a methyl group takes place from S-adenosyl methionine, abbreviated as SAM, and it is given to the substrate. Now, the reaction in this way proceeds. Now, if we look at this diagram, we can see that the adometh group is as such attached to the positively charged sulfur atom, due to which it makes it highly reactive towards the nucleophiles like oxygen, nitrogen and activated carbon. Otherwise, the methylthiol group is relatively inert. As a result, the SN2 reaction needs to be proceeded forward, so it must be catalyzed with the help of certain charge distribution. Now let's move further on. So let's see what are the different types of methyltransferases occurring in prokaryotic system. There are three different types of methyltransferase enzyme. The first one is known as M6A, the second one is known as M4C, and the third one is M5C. The M6A suggest for those kinds of methyltransferase enzyme that catalyze the transfer of a methyl group to the amino group present at 6th position of adenine. Similarly, the enzyme which is abbreviated as M4C, that is the enzyme which catalyzes the transfer of a methyl group to the amino group present at carbon 4 of cytosine. Whereas the M5C methyltransferase catalyzes the transfer of methyl group to position 5 of cytosine. So as we can see, since there is a transfer of methyl group, so there is a change in the structure of bases, and hence the bases are becoming modified. We know in DNA there are 4 bases: adenine, thymine, guanine and cytosine. But out of 4, only 2, namely adenine and cytosine, are capable of accepting the methyl group, and hence they are becoming modified. Let's see the structure of these modified bases. Adenine, after taking in methyl group, changes into N6-methyladenine, whereas cytosine can accept methyl group at two positions, the position 4 and position 5. The position 4 then becomes N4-methyl cytosine, whereas the position 5 becomes C5-methyl cytosine. So in this way, we can see how the modified bases of adenine and cytosine are formed after accepting the methyl group from S-adenosyl methionine. After understanding about the different functions or what are DNA modifying enzymes, what kind of bases are modified, now let us see that how does it help or what is the function of these DNA methyltransferases. The DNA methyltransferases, it creates modified bases. Now, with the help of these modified bases, it is able to differentiate between the two strands of DNA during DNA replication, during your DNA repair system. The third important point is, with the help of these modified bases, the conjugal transfer of plasmids is dependent. The fourth important point is, it plays a very important role in controlling the bacterial pathogenesis and hence virulence. Now let us move what is the importance of methyltransferases in recombinant DNA technology. The methyltransferases came into focus much more with the advent of RDT. It was found that cloned DNA, which were first cloned in bacterial cell expressing DAM and DCM, makes the plasmid DNA resistant to endonuclease cleavage. So after knowing about the functions, now let us come to the first type of methyltransferases commonly occurring in prokaryotes, that is DAM. The DAM stands for DNA adenine methyltransferase. So this group of enzymes catalyzes the transfer of a methyl group to the adenine residue. Let us now see what are the different physiological functions which are catalyzed or done by DAM. Now, before coming into the details of all this, we first need to know that what happens during DNA replication. Now, DNA replication, we know in both prokaryotic and eukaryotic, it belongs to the semi-conservative mode of replication. In this, the old strand or one of the old strand is acting as a template and another new strand is synthesized. Now, during this, what is happening is, after the DNA has replicated, the old strand contains methylated bases. The newly synthesized strand, after a short while, also contains methylated bases. Now, just after the DNA replication, the newly synthesized strand is devoid of methylated bases. And it helps in understanding the different strand, the parent strand as well as the newly synthesized strand. Or in other words, the hemi-methylated state of DNA molecule plays a very important role.
Ma'am, I have got a question. What is the importance of hemi-methylated states in prokaryotes?
First, let us come to this term, hemi-methylated state. As the name suggests, hemi means 50% methylation it's been done. As we know, their DNA contains two strands. So in this case, one is methylated and one is not. And this state is known as the hemi-methylated state, or the state in which only 50% of methylation of bases has been done. The hemi-methylated state plays a very important role in differentiating the old strand and the newly synthesized strand, because during DNA replication, the newly synthesized strand might contain a few misfolded or mispaired base pairs. So it helps in distinguishing the newly synthesized strand. Now let us see how DAM helps in replication. We know that the timing of the cell cycle needs to be coordinated. Bacterial cells divide very rapidly, and each bacterial cell must contain only one nucleoid or only one copy of the nucleoid. If the DNA replication occurs too fast in comparison to bacterial cell division, then non-viable cells will be formed, because some of the cells might not receive the duplicated DNA copy. On the other hand, if the DNA duplication occurs too fast in comparison to cell division, then one or few cells might contain more than one copy of nucleoid. So the bacterial cell division needs to be coordinated very well or coordinated tightly with the DNA replication. And this is done with the help of DNA modifying enzymes, mainly the DAM enzyme. Now let us see how it is done. So, if we remember, during the replication we have studied that the initiation of replication takes place only at certain specific regions along the DNA molecule, which is known as its origin or ORIC. If you closely examine the DNA bases which is present at ORIC, you'll find the sequence GATC. Now, in the sequence GATC, there are adenine residue. And it is this adenine residue which is methylated. So as shown in this diagram, we can see that the GATC site which is present in ORIC, so it is becoming methylated as shown by the asterisk mark. During DNA replication, now what is happening? The newly synthesized DNA strand does not contain any methylated bases, and hence it is present in hemi-methylated state. Now just after the DNA replication, immediately after the DNA replication, so we get the hemi-methylated state. The competitors for this hemi-methylated state are Sec-A and Mut-H protein. Now how does it help? The Sec-A protein or the Sec term stands for sequestration protein. Now since it binds to the hemi-methylated DNA, DAM enzyme is not able to methylate the bases present in the newly synthesized strand. Now as a result, there is a delay in DNA replication, in the next round of DNA replication. Now since there is a delay in the next round of DNA replication, within which time the bacterial cell can divide, and so that there are equal number of copies going to the daughter cells. So in this way, as shown, we can see that the Sec-A is we are inhibiting the binding of DAM enzyme. On the next half, we have here, after the Sec-A is released, then DAM is able to methylate the adenine residues present on the newly synthesized strand. Now let us see how the Sec-A protein helps in membrane sequestration. So as I told you, immediately after DNA replication, hemimethylation state is there. Sec-A protein is binding to the hemimethylated state or hemimethylated DNA, and thereby the enzyme such as your DAM enzyme is not able to act to it. Now after it has the hemimethylated DNA, after it has bounded or formed a bond with Sec-A, it binds with the bacterial cell membrane, and as a result the DNA becomes more compact and nucleoid formation takes place, and it helps in cell division. So you can see in this diagram, there is a Sec-A protein which is binding to the hemimethylated DNA, after which it is binding to the cell membrane, and then what is happening, since it is now bound to the cell membrane, next round of DNA replication cannot take place. There is another protein which is known as DNA protein. Now since it is not getting the template, that is the fully methylated DNA, as a result it is not able to catalyze immediately another round of replication, DNA replication. So in this way, the hemimethylated DNA stays in the hemimethylated form for up to 1/3 of the cell cycle. After which what happens, the levels of DNA present in the cell declines, and then the Sec-A is released from the hemimethylated state, and now DAM can methylate the adenine residues. Now, since DNA A is not available, so it cannot catalyze immediately another round of replication. So in this way, the DNA replication is tightly coordinated with the bacterial cell division so that each daughter cell is able to receive only one copy of bacterial genome. So in this regard, the DAM enzyme plays a very important role. Now let us move to the second important physiological function of DAM. DAM plays a very important role during DNA repair system. During DNA replication, an incorrect base pair or mismatch base pair might be incorporated by DNA polymerase III. Before the cell goes for cell division, this kind of mismatch or incorrect base pair needs to be removed from the DNA. In this regard, the hemi-methylated state plays an important role. Now let us see what are the types of repair system in prokaryotes. The mismatch repair system is removed by two types of enzymes. One is a nucleotide excision repair system, another is the mismatch repair system. The mismatch repair system, it requires the presence of three proteins, MUT-H, S and L, which are encoded by MUT-H, S and L genes respectively. To understand better how these proteins are helpful in your taking away the incorrect base pair, let us look at this diagram. Now, as shown in this diagram, there is a mismatch between G and T. As we already know that G base pairs with C. So, by incidence or due to a certain fault in DNA polymerase III, a T has been incorporated in place of C. So, it needs to be removed. So what happens? The first protein is the MUT-S protein, which recognizes this mismatch base pair and binds to it. The MUT-H binds to the two sides of the MUT-S protein and it is able to differentiate or locate the nearest GATC site or the modified adenine present in the GATC site adjacent to it.
Ma'am, one question. How does MUT-H understand which is the daughter strand?
The MUT-H is able to differentiate which one is the daughter strand by the presence of hemi-methylation state. Now, as we know, MUT-H is able to recognize the methylated adenine present in the nearest GATC site. Now, as a result of binding to the adenine residue, it is able to differentiate which is the newly synthesized strand, because the newly synthesized strand is devoid of any methylated adenine. And therefore, it can make a cut in the newly synthesized DNA. So, in this way, it is able to differentiate which one is the daughter strand. Now, what as I was talking about, the MUT-S first binds, MUT-S and then MUT-H binds. The MUT-H, after the binding of MUT-H, it recognizes the modified adenine, followed by the addition of the MUT-L. So, MUT-SHL forms a complex and then the DNA is pulled through this complex in the form of a loop. With the help of modified adenine, MUT-H is able to differentiate the newly synthesized strand and it makes a nick in the newly synthesized strand because the incorrect base pair is present in the newly synthesized strand. The strand is then removed and the repair or the gap is repaired by the DNA pol III using the old strand as the template and the correct base pair is incorporated. So, in this way, the hemi-methylated state or rather the modified adenine helps in distinguishing between the old strand and the new strand. So, this is one of the most important or vital physiological function of DAM enzyme. Let us just see in a glimpse or in a short view how DAM methylation can also help in regulating virulence. This is an example of Salmonella species. The Salmonella species, it has been found that DAM is absent, such as DAM mutant Salmonella. There is an distribution or there is a change in its virulence properties. As a result, you can see that it displays altered morphological structure. Like there is a distorted outer membrane, the proteins are leaked out from the DAM mutants. It is not able to produce proper pili and as a result the pathogenicity island is also altered because of absence of DAM, and as a result it is not able to establish infection in the host immune system.
Ma'am, what is the function of DCM?
The function of DCM is, first let us see what is the full form of DCM. DCM stands for DNA Cytosine Methyltransferase. Again, similar to DAM, it is a modifying enzyme and thereby it helps in creating the hemi-methylated state. The hemi-methylated state as a result helps in distinguishing between the old strand and the new strand, as well as it plays a very important role in DNA repair system. So these are same for both DAM and DCM. The only difference is in the structure of DCM, and the DCM is most commonly found in the eukaryotic system rather than the prokaryotes. So in this way, we can see that the DCM is one of the important modifying enzyme present in the eukaryotes. Now just let us see how does the structure of DCM look like. The DCM is made up of two domains. One is the large domain, subdomain, that is the subdomain is a part of the substructure of protein or enzyme, and a small subdomain. The large subdomain binds with the adometh and the substrate binding ability, and whereas the small domain you were able to recognize the sequence, that is the cytosine in the substrate domain. Now another important function of your 5-methyl cytosine generated as a reaction of DCM enzyme is that it helps in the VSP repair system. The VSP is very short patch repair. Now very short patch repair is necessary because the 5-methyl cytosine sometimes becomes deaminated and thereby generates thymine. Again, thymine has got, because of the generation of thymine, the base property or the base pairing becomes altered and becomes in mismatch base pair. The mismatch base pair is removed by the help of very short patch repair system. In this, the DNA region adjacent to T is first cut and the fragment is removed, and then the fragment is repaired, or it is repaired with the help of DNA polymerase 1, and the gap is sealed by the ligase. So in this way it helps in the removal of T generated from the deamination of 5-methyl cytosine. In eukaryotes, the CG sequences are present in specific part of the DNA which is known as CPG sequences, and they serve as the hotspots. So in this way, both DAM and DCM are important modifying enzymes present in the living world.